Hello, and welcome to Let's Talk SADS Live, Canada. I am your host, Dr. Shu Sanatani, and delighted to be coming to you on our 20th episode. Hard to believe, and coming to you this morning from Vancouver. But I know you're tuning in from all over the world, and some of you are watching this later in the quiet time of your day. Really excited to be part of this series where we learn together about SAD, sudden arrhythmia, death syndromes, conditions, inherited heart rhythm conditions, often running in families, sometimes presenting with very impactful or tragic events. And we are learning about these conditions, which are generally rare, especially as the usual parlance of medicine goes. It's not like the diabetes or sore back that everybody is aware of. So we like to make sure that we have a good, healthy dialogue to inform our audience, many of whom have been impacted by inherited heart rhythm conditions or SADS conditions themselves. So thank you for joining. Excited about our show today. And a few things that I like to do before we invite our guests in. And let's just share the screen here. Thank you to Evan, our Director of Communications, who makes everything possible. And so here we are. You see gene therapy and CPVT. So more about that in a moment. And I just want to talk a little bit about something that was going on earlier this month. The Hearts and Rhythm organization, of course, close friends and partners of ours, who really are a national network of inherited heart rhythm specialists, healthcare providers, industry representatives, patient advocacy groups, first responder, really a diverse network across the country, and close partners with the Canadian SADS organization and HERO, as we abbreviate them. They have an annual meeting, and we got together in Toronto. I want to say last week. The June is just running by like a blur, and had some amazing discussion. And you can see there actually a panel, including Evan and myself, and our speaker, one of our guests today, Dr. Gabe Brooks. The meeting itself was really something from the future, from my perspective, where so many of our new learnings were brought to light. And here's our SADS team with the organizers, and of course our executive director, Pam Husband, who had the vision to start the Canadian chapter of SADS many years ago. I won't age either of us, but it's been a long time. And our board president, Nadia Larson, joined us as well, and Brianne Davies, who of course is the heart and soul of the HERO organization, founded by Dr. Andrew Cron, who has been a guest here. So wonderful meeting. I'm just going to put this bright red. This is a very jarring slide, but just to remind us that even though Canadian SADS interacts regularly with industry and other financially active organizations, our views are really, we do not necessarily endorse a specific treatment or organization. We are really here to disseminate information and to sift through information, relying on scientific methods. So we also cannot necessarily manage your own personal case. We tend to be able to give you guiding principles. So the talks are still available. Some of them are still available on our YouTube channel, I think. So if you can scan this QR code, you can still access some of the talks. And I don't actually know how many of the talks are still up or how easy this is to do. But Evan might put in our group chat, where you can also submit your comments. He may put a little more information about that if he has time. But yeah, try scanning the QR code. Now, the other thing you know me to do is I like to have a little bit of fun facts and and so just a reminder, we talked early on about CPVT looking like digitalis, toxicity, digoxin, of course, is a plant. Some of the origin of arrhythmogenic cardiomyopathy, Brugada, the first series, again, some facts about arrhythmogenic cardiomyopathy. And then last few, we talked about long QT syndrome, including that the ECG really wasn't recorded until about 100 years after people suspected the syndrome. And the interesting association between the hearing loss that some cases, some patients have, particularly with severe long QT syndrome. So what do we have today for a did you know that? Well, today I went a little bit along the theme of our talk today. And this is as much a surprise to me as it may be to you, that we have been reading about gene therapy for over 50 years. And this is just a couple of excerpts from this really a landmark article in the journal Science 50 years ago, Gene Therapy for Human Genetic Disease. And this figure actually shows virus particles being brought into a cell and integrating the DNA that the virus is carrying. So while we may feel that gene therapy is cutting edge and just has fallen out of the sky, there's actually a really interesting story and history behind it. So importantly, this article also commented really that gene therapy was not ready for prime time at that time, largely due to ethical and other concerns. So so really excited today to introduce our guests. So Dr. Gabriel Brooks and Ms. Annie Ganot, both who are with Solid Biosciences, and I'll just do a little bit of an intro to them. Sorry, here I'm just fumbling around. So Dr. Brooks, he's a trained clinical cardiologist and the chief medical officer at Solid Biosciences. And I'm going to let him tell us a bit more about his background. And Annie is a co-founder and the head of patient advocacy at Solid Biosciences. But rather than me reading off their very impressive bios, let's bring them into the studio and start our conversation.
Hi, Shu. Thanks so much for having us and for hosting, I think, this extraordinary broadcast. And we're just really pleased to be here.
Well, thank you both so much. I'm going to start with a formal welcome, Dr. Brooks, and it will probably digress to a Gabe or Gabriel. And similarly, Ms. Ganot, it will likely degenerate to an Annie, and please call me Shu. I will, before we get started, I will disclose that I have served as a medical advisor in a limited capacity for Solid Biosciences. That I don't think will impact our content today. And certainly Solid is one of many important companies that are working to improve the care of our SADS patients. Let's start with you, Annie. Tell us a bit about how it is that you are here as a co-founder of a biotech startup company.
Sure. Thank you so much for having us, Shu. We really appreciate it. We're excited to have the opportunity to connect with patients and caregivers. We founded Solid Biosciences about 10 years ago, following my son's diagnosis of Duchenne muscular dystrophy. When our son was diagnosed, my husband and I were living in London and were not in the biotech or pharma space whatsoever. And we looked around the landscape and decided that we hoped that by starting a company and surrounding ourselves with the best and brightest in Duchenne muscular dystrophy and ultimately gene therapy, that we may have the opportunity to try to move the needle for patients in his lifetime. So that was really the genesis of Solid. So Solid is based in Charlestown, Massachusetts. We've been going since about 2014, really focused on both neuromuscular and more recently cardiac indications, and very much focused on gene therapy. So that's the background to the company and my personal story.
Yes, and thank you for sharing that. For our audience who may not be as familiar, Duchenne muscular dystrophy is what's called X-linked. So it's a sex-related condition primarily affecting males, almost exclusively affecting males, and a lot of muscle involvement, and also the heart. And the protein involved is actually one of the larger proteins. So the fact that gene therapy is actually in clinical trials now is really, really amazing. So um, but today we're not going to focus on that, but we're very grateful that um, Annie is here with that lived experience and that perspective. And Dr. Brooks, Gabe Brooks, you come at this from a different lens. You're a clinician.
That's right, Shu. Thanks so much. I am a clinically trained cardiologist, as you mentioned, but I've been in the biopharma space for a number of years now, in large and small companies, and had worked on drugs, small molecule drugs for arrhythmia. But the last 10 years been dedicated really to this idea, this this proposition that precision medicines that really target the disease, what's the fundament of the disease, may be able to better have a clinical effect. And we're using molecular genetic tools in this setting, gene therapy, to affect that. I joined Solid back in October, and I just want to lean into a little bit of what you said, Shu. I think Solid Biosciences, having really been in the space of Duchenne muscular dystrophy, a really tough disease for a number of years, really allows us as a company to understand the importance of listening to the patient, what's important to them, and really trying to hone our approach in that way. So it's really a privilege to be here on their behalf, and to be here with you today.
Well, and thank you both. And you both attended the Hero meeting, and it was nice to interact in person. That's still on the to-do list for the Canadian SADS team to get us into an in-person meeting. In the interim, though, we'll really take a leadership role in educating our patients and making sure they have access to good information. So the concept of gene therapy is sort of this massive term in two simple words. I'm still astounded. I didn't graduate from medical school that long ago, but I didn't necessarily think that it would be within my career that we would be talking about gene therapy for heart conditions. So maybe Gabe, just for our audience, just take us back a little bit about what is the basic concept here. So I think our audience understands that several of our SADS conditions are inherited. So there's a spelling mistake that we can identify in the genetic code. How do we go from that to actually the therapy part?
Absolutely. So Shu, gene therapy is actually a pretty broad term, and encompasses many different approaches. Some of the approaches that you were referring to use something that's called a lentivirus. It's not really important, but it will actually, through that approach, there will be physical changes to the DNA, physical changes to really the blueprint of the body, to make sure that spelling mistake is corrected. That's one type of gene therapy. On the other hand, there's another type of gene therapy, and it's the gene therapy that we're using that doesn't integrate into the DNA. So it doesn't actually change the DNA of the patient, but what it does is that it expresses in the nucleus. There is a piece of DNA called a transgene that sits there in a circular form that's that enables an expression of a protein in the cell itself. And it's really that protein that's the therapeutic. And so that's a different type of non-integrating gene therapy. And in this case, that we are prosecuting at Solid Biosciences, it uses what's called an AAV vector. And I'd love to share kind of the concepts of what that means, the transgene and the vector, in terms of a therapeutic approach as well.
Yeah, so fantastic science that one can actually sort of bring in a piece of DNA. DNA, of course, the main purpose of that is to provide instruction to make proteins, and the body, all the bodily functions are carried out by proteins essentially, and different forms of the protein. So to be able to bring in a correct blueprint to make some protein that helps the defective protein is really, I think, the essence of it. And I learned at the conference that there are so many different approaches to that now. Annie, what do you like about the direction that Solid is taking? Again, not to go into proprietary things, but as a patient advocate, and as a maybe even more more invested than some of us in the science side of it. What are some of the things that excite you about what you're doing?
I think it's a great question. I think if I go back 8-9 years ago, and we as a company wanted to go forward with gene therapy as a potential treatment option for Duchenne, which, as people and everybody starts to understand more and more about how complex, certainly Duchenne is such a complex disease, affecting all skeletal muscle and cardiac muscle, and to be able to deliver in such high doses. I just remember sitting in a room with, everybody, so many clinicians that understood, and scientists that understood Duchenne and understood gene therapy. And they were like, if you're going to go after any disease, like let's go after the liver or go after an eye or something that's much smaller and more manageable. Like if there was a list of 100 diseases that you were going to use gene therapy as a potential modality and treatment option, Duchenne wouldn't ever feature on that top 100 list. But myself and my husband were like, well, our problem is Duchenne. So let's figure it out. Like, let's put your egos aside. Let's put your desire to publish papers aside, and let's figure out how do we safely and collectively get into the clinic for Duchenne with using gene therapy. So I think, in a long-winded way of answering your question, there's so much that we've learned, having been in the clinic with a gene therapy, an AAV gene therapy approach for Duchenne muscular dystrophy. There's so much that we have learned, and the families that have participated are true heroes in every sense of the word. And now the beauty is that we get to take all those learnings from a manufacturing perspective, which that's a beast, understanding delivery, understanding, looking at immune suppression, every piece that has to go into it, we have learned, and very humble about that approach, and making sure that we do this in such a thoughtful way that now when we look at applying gene therapy for other indications, and certainly for cardiac indications, I feel like that foundation of understanding and true lived experience in that space really helps set us up in a very thoughtful, professional, and intelligent way.
Yeah, thank you. Certainly Duchenne is a challenging condition, multi-organ involvement, really, and that that is remarkable. Now, so CPVT, just to remind our viewers, catecholaminergic polymorphic ventricular tachycardia. The essential issue is that adrenaline brings out an excess of calcium in the cell that causes these lethal rhythm disturbances or arrhythmias. So emotional excitement, less so planned exercise, but sometimes relatively usual activities of daily living. Depending on the other triggers, the average age is around 10 years of age. So CPVT is an interesting choice to target. Gabe, tell us a bit about why CPVT. It's 1 in 10,000. It's not, it's generally well managed with medications. What's the rationale?
Thank you, Shu. So I think our first approach when we're thinking about indications is really to listen to listen to the community and listen to the patient. And, through that, what we've learned is that, yes, there are medicines that that can, if taken, really assiduously every day, high dose beta blockers and flecainide can really prevent most cases of an event. But still, we're having events in the community, and those events, when they happen, can be lethal. And so, we want to be able to do better for those patients and to liberate them from their fear. Sometimes patients have what are called implantable cardiac defibrillators. I'm sure a lot of your viewers are very familiar with that device, and that can be life-saving, but it also can cause pain and shock and psychological stress. So if we had a therapy, and we're thinking about these patients and their families, and this burden of, if an event happens, what will it be? When will it happen? Are there ways to prevent it? If we have a therapy that could provide a continuous, durable solution to that protection, and I will just say here now, I'm not making any claims about our medicine, because this is the goal of the medicine, but our medicine, we still need to investigate in clinical trials. But that's the proposition. If we can provide, if we can find a medicine that can provide continuous durable therapy to liberate that patient and their family from fear, and potentially to allow that patient to exercise without fear, or be exposed to emotion without fear, and live a full life, then that's meritorious. That's something that we want to do and be a part of. So for us, we're very passionate that CPVT needs better solutions, and we'd love to talk about kind of our approach to that disease.
And I think we heard it loud and clear. A shout out to our neighbors to the south, the SADS US Foundation. They sponsored another session with the FDA earlier this month, where hundreds of people provided support for the need for better care, and that I think is still also available to listen. Alice Lara and Mike Ackerman have done an incredible job getting the patient story in front of the FDA about long QT and CPVT specifically. So completely agree. And I'm always I'm walking two sides of the fence. On the one hand, I'm very reassuring with my patients about the safety of doing things and the importance of taking their medication, and while recognizing that this is not a benign task, and it's not a trivial burden for these teenagers to take usually 3 pills a day, and to have a safety plan, and many of them have these implanted devices or have access to automated devices. So there's definitely in our future, we definitely want a better treatment for sure. So maybe just give us, you said you'd like to give us a high level view of your approach. Please give us that, so we understand sort of your not the specifics of your secret ingredient, the general principles, because I'd like to follow up then, so our viewers can understand, what does it actually mean to potentially go into a trial?
Absolutely. Thank you, Shu. So the concept of AAV gene therapy is really this. There are really 2 components to the drug. One is the adeno-associated virus. Now, an adeno-associated virus is a benign virus that exists in nature that we are exposed to, and it doesn't replicate. It's not able to produce other virus itself, unless there's an infection with an adenovirus, and an adenovirus is a typical cold virus. And so this virus just kind of hangs around, and it doesn't really have any known pathology when it infects patients. But what it does is that it's evolved over hundreds of millions of years to be able to get into cells and to get its genetic material into the nucleus of the cell, so that if there was an adenovirus, it could then replicate. And so how does it do that? Well, it has this shell. And the shell has the instructions that we still are trying to understand exactly how it works. But in a basic sense, we can think of it as a delivery truck, like a UPS truck, and or a package, if you will, and that has an address on it. So it knows to go to the cardiomyocyte, just like that package knows to go to my house if I've ordered something from Amazon. Then what's really important is not that we get the package to my house, but what's in the package. Normally in an adeno-associated virus, there are the genetic code for itself. But what we've done is we've taken out that genetic code and we've put in a new genetic code for a protein that we think is important for treating the particular disease. So if this was something like a long QT syndrome, it might be genetic code for, KCNQ1 or KCNH2, which is long QT1 or 2, or even SCN5A. There are, of course, a lot of complications about how big that genetic code is. But I'm just trying to convey the sense that we can tailor that message for the particular protein that we want to express. In our case, we're really interested in stabilizing the ryanodine receptor, which is important in terms of its diastolic leak, in CPVT 1 and 2. And so we've put in the genetic code for a protein called calsequestrin to try to stabilize calcium so you don't have abnormal leak of that ryanodine receptor that causes the manifestations of the abnormal arrhythmia in CPVT 1 and 2. So Shu, just to recapitulate very simply, adeno-associated virus, two parts. One is the package that ships the genetic code to the right place. In this case, it's the cardiomyocyte. And that's that adenovirus vector shell. Adeno-associated viral vector shell. And the other is the genetic code, which is the package, the stuff on the inside. And in this case, for our approach, it happens to be calsequestrin.
And a very interesting approach. And I know others have initially focused on a different type of CPVT where calsequestrin was the abnormality. Ryanodine, of course, is this huge protein and it relies on a lot of other molecules for it to work properly. So using basically a partner protein that can get into the cell is very creative and innovative. And I do want to acknowledge the incredible work of Silvia Priori, who has done so much of the background science, and other sort of visionary in our field who remains years ahead of where the clinical field is. So she's really enabled a lot of this work. So one can't just come into my office with a vial and a syringe and say, okay, let's inject the next couple of CPVT patients. So, Annie, are you able to sort of speak a bit to the regulatory processes and how to set up the trial, or is that something you both want to tackle? What does it actually look like? So from getting a center going to finding the patients to taking them through.
I'm going to let Gabe kick that off, and then I'm happy to pipe in.
That's great. And Annie and I really work in close partnership here, because there's the scientific aspect of making sure that we have set up the right experiment. But goodness, I don't care how well set up that experiment is. If you relationships with patients and families, then it's not going to go anywhere. So this is a good two-part question, I think, or two-person question. So Shu, the most important thing of any clinical design is that we understand the safety of the product. And so we do a lot of work even before we start step in your door to ask for your partnership, for instance, in being a participating center, let's say. And I'm not saying that I have or anything like that. I'm just making the example. We need to create a dossier to go in front of the Canadian regulatory agency in your case. And that has to show initial safety in animal studies and biochemical assays and the rest of it. So we really need to create an entire package that says that, and go in front of the regulators and go in front of you and say, to the best of our ability, we've interrogated all aspects of this drug, and without dosing any humans, we think that this is as safe as we can estimate by whatever methods we can do. But then, the first thing is it safe in people? So we need to partner with brave participants and brave families who are engaged in the proposition of saying, you know what, I think that we need to learn more about this disease, and maybe learning about this drug will help us understand what, that this therapy potentially might be effective. The next thing is efficacy. So we need to design, listening to folks like yourself, what are the best efficacy, and efficacy means the clinical impact of the vector of the medicine. And so we think very carefully about that and try to, again, in partnership with the patients, understand what they can do, what's going to be acceptable for them. In our case, for CPVT, it's obviously looking for causes of arrhythmia, and do we see any decrease in the arrhythmia using this drug? So that's the kind of basic concepts. Initial study, focus on safety, make sure that we can proceed in a safe way with this medicine and learn as much as we can. And certainly we've done that with all of the other gene therapies that are out there for different diseases, and we're certainly learning about that. And then we can think about, how much is this going to actually help the patient in terms of decreasing arrhythmia.
And I think just to layer onto what Gabe just explained, I think certainly from the very earliest time point, if you have some good science that as a biotech company that you license in, at the very earliest point, we have to ensure that we have a good relationship with the patients and the caregivers and understand what matters to them. How do they feel about their current medicines that they're taking? What would be appealing to them? Truly understanding, that whole benefit risk profile, what would they be willing to go through, and what to them is success, right? I think that we, as a lot of companies, think that they know what the success is, but until you really truly, connect with patients and caregivers and understand what matters to them most, I just think it's a disservice to the community. So that's a big part of it. So even at this point for Solid, we have been engaging with the CPVT community, holding focus groups, reviewing things like our protocol design. Is this something that you think that a patient could go through? Does this make sense when we look at the assessments? Do these look like the right endpoints? Are we measuring the right endpoints? So really trying to understand and get that constant feedback is critical to success. Even before you're going to the regulators, even before you're going to the agencies, whether it be in Health Canada or here with the FDA, to truly understand what we're proposing not only makes sense scientifically, clinically, but that ultimately it's what matters to patients and families.
Well, thank you both. Really a sort of a 360 view of the complexity. And in the SAD space, in the inherited arrhythmia space, we often talk about shared decision making. It sort of attached itself to athletic participation. But really, this epitomizes that, doesn't it? Where our responsibility is to ensure that we have very good dialogue, and then these patient partners are going to go away and think about it. And I like Gabe's use of the term brave, because the first few iterations are not necessarily going to be the final right dosing, or they might be dose finding. You might even have some people, depending on the methodology, there might be people who are in a placebo arm at first, if we're going to be really rigorous scientifically, to make sure people don't just improve on the imagination of a medication. So lots and lots of commitment from those first patients, and I have to wonder if, because CPVT is largely an inherited, often familial condition, some of these early participants or volunteers are going to potentially be doing it for their children, the generations to come. And I'm reminded of the here in British Columbia in particular, where the health organization I work for is really learning from our Indigenous partners, and talking really about 7 generations of impact. And one has to think that really these first few people they're really thinking about participating to improve the care of CPVT, that may not even benefit them directly. Not to, I mean, it might be, we don't know exactly, how well things are going to work. The science is very solid, pun absolutely intended. But it might be that the grandchild now has a totally new paradigm of treatment. So it's just really important stuff to think through. And I'm glad that your company and all the companies I've interacted with really are putting the patient at the center.
That's right. And I think when we are coming to the clinic, meaning we're bringing a drug for clinical testing and going to sites and going to patients, we only do so if we feel there's tremendous promise, and if we have confidence in the rationale of the drug that we are bringing to them. But also we need to come in with a huge amount of humility, around making sure we're watching everything like a hawk for safety, and that we're really listening to the patients about, how is this affecting you? How can this help you? And make sure to measure those things in ways that will be meaningful.
So let me ask you again, both a harder question, or maybe an easy question. What are some of the things that keep you up at night? Some of your fears, some of your fears around what you're embarking on. Maybe Annie, start us off.
I'm happy to start. I think certainly one aspect of gene therapy as of today is that it is a one-time therapy in this moment. And so when we speak about these heroes and these brave, children, adults, families that are participating in these clinical studies, we do not have the opportunity or the option right now to redose. And so getting it right the first time, making sure that dose is of course safe, but on top of that is efficacious, is critically important. I'm certainly applying these learnings with Duchenne and now for cardiac indications, but truly, that's what keeps me up at night, is that, we want to make sure that we are absolutely doing the very best that we can. And Shu, your point earlier, where we are with the science, I don't think we're flying the Wright brothers airplanes anymore, but we're certainly not going to the moon, right? So we're still in that earlier phase of understanding and clinical experience in patients, but we're not there yet. So I was kind of cautiously optimistic about this, and just making sure that families truly understand what they will be getting into from every aspect.
I'm going to lean into a really good point that Annie, I think, just made, which is what we call the minimally effective dose, which means the dose that we're starting in the clinic in gene therapy. And in contradistinction with things like small molecules, where they can last a couple hours or maybe a day or two, and then they're out of your system. We in pharma, we usually start at a very low dose and we increase the dose over time in usually healthy patients or healthy participants to study the safety of that drug. Gene therapy, when we are exposed to an adeno-associated virus, we're exposed to a couple of particles very much. When we give it therapeutically, we give a very large dose. And the immune system, this is something it's not seen before, and it makes what are called antibodies. And those antibodies clear out the virus the next time. So we're unable, if we give a dose of a gene therapy, we're really unable to ever give a second dose to that patient today. There's lots of work on technology to overcome this challenge, but right now it's a pretty big challenge, and we don't really have a solution really in sight today. And so this paradigm of starting from a minimal dose and looking at safety and getting up to what we think would be an effective dose is something we can't use in gene therapy. We have one shot in terms of that patient. And so we need to start, if we're going to expose patients that have a disease that we want to be able to provide a therapy, we can't start at a dose that's low and feel comfortable because we think it's a low dose and safe, because then they'll never, ever be able to get a therapeutic dose. So we really need to go thread the needle where we think, gosh, this is a safe dose, we think, based on all of the data that we have, but it gives us the potential of actually having an effect on this patient. So that is that is a big challenge that we work through all the time.
And it's in our current era, post-COVID, we have so many more questions about the immune system, perhaps, than we had even before. So I'm sure that some people, and we want to make sure that people get their information from credible sources. But there were so many experts on the immune system overnight. So importantly, though, I think as we have learned, there's also hope in the future that we will be able to work with the immune system and potentially, make the immune system work a little bit with us, plasmapheresis, things like that, well beyond my scope of expertise. So just in our last sort of moment or two, if you could each just give us where are we in 5 years, or flip it, turn it around and say, viewers at home, don't expect this to come to you in the next 6 months, but so either tell me your time horizon for your own venture, or generally where you think we are in 5 years. Gabe, we'll put you on the spot first.
All right, great. So I think first, there's a lot of really thoughtful and dynamic companies in this space working on different gene therapy approaches to some of the diseases that touch SADS community, and even those that aren't necessarily directly SADS related, that are cardiac related. I think over the course of these next 5 years, we're going to learn so much about how to develop precision molecular genetic therapies for rare cardiomyopathies and inherited arrhythmia syndromes that we don't know today. So I think that the prospect is good. I'll say one thing, sorry, as a digression. For a long time, gene therapy was the future of medicine, but not yet here. It's here. There are gene therapies that patients can get through insurance, through commercial sources for diseases like spinal muscular atrophy, and now Duchenne muscular dystrophy, not our company, but another company. So it's no longer the future, it's here. However, we have so much work to do, but we have a lot of companies that are engaged in really important cardiac indications. So I think we're going to learn a lot. For us, we've shared that we're really looking forward to opening up a study with a gene therapy for CPVT in the next year. And so we really would look forward to being able to talk to the community about what this study would look like, and to again listen to them in terms of, how can we best serve them as a partner.
Annie, your comments?
I think for patients and caregivers, I think it's never soon enough. So I'm going to just start with that. It's never quick enough. However, as Gabe said, we are in the space right now where gene therapy is in the clinic and treating patients, not only in the clinic, but also commercially available for some patients in some populations. I think that the exciting thing is that we are learning so much, and in that partnership with advocacy groups, SADS Canada, SADS in the US, that partnership is going to be critical for our success. So I can't emphasize enough that, as we gather questions and concerns from the patient community, that really helps us be so much better and smarter and more thoughtful about everything that we do. So I think, from my perspective, I'm just really excited to be continuing that partnership and working very closely with SADS Canada, and also clinicians and everybody in the space to make sure that we collaborate and that we do this in partnership and do it well. And the patients are at the very, very center of everything that we do.
Well, thank you both so much for joining. And I feel like we could have gone off certainly into a lot more conversation about this. This is here and amongst us, living amongst us, and we are actively learning. And so thank you both for your time, and thank you also to Solid for really putting yourselves out there to improve the lives of our patients. So we will look forward to hearing more about this, and our patients will definitely look to companies like yours for hope for generations to come. So thanks very much.
Thank you so much, Shu.
Thank you. Thank you for having us. Thank you.
Really interesting stuff. And, and we saw at the Hero conference, this is just one approach. A lot of technology is really aligning to open up new and novel approaches to our SADS conditions. So we are actually a little bit over time today, so we're gonna call it a day. Summer is going to start this week, and I will look forward to seeing you over the summer. And thank you for your support. Consider making a donation. It makes it possible to have these webcasts. It also pays our office and keeps the lights on, so that we can keep supporting our families. Thanks very much.